Thank you for coming. We want to start the session today from, we start at 4 o'clock, and this is the second session that we have general discussions on COVID-19 vaccines. The first session was last Thursday regarding the mechanisms of vaccine development, or what is called vaccine platforms. And today, Forrest Arnold, another faculty member from the ID division, is going to give us an overview of the phases for the vaccine studies. And as we did the last time, you can type your questions, and he probably, Forrest is going to talk for 20, 25 minutes, 30 minutes, and then we're going to go another 30 minutes for questions and answers. And then Ruth Carrico is in a meeting. She cannot attend today, but this coming Friday, Ruth is going to talk about safety, vaccines safety. Then Forrest.
Thank you. I'm glad that you all have joined today, and we hope to share you some good information about vaccine trials in general, and appreciate you dialing in. The outline for my talk today, I'm going to talk about the FDA for a moment and then go through the different phases of a trial, and then talk about Operation Warp Speed, which is what has been termed this COVID-19 vaccine. So first of all, the Food and Drug Administration has 6 centers. One of them is the Food and Safety Applied Nutrition. That's where cosmetics go as well. Then there's the Drug Evaluation and Research, which seems like a good place for drugs and vaccines, but it's not. Then there's Veterinary Medicine, Tobacco Products, Devices, and Radiological Health, as well as Biological Evaluation and Research, and that's the one where vaccines fall. And so this center of the FDA is in charge of vaccines. What you see, and this is the best figure I could find, was that the very first thing to happen with research is basic research. So these are people in benchside laboratories, and they're doing their research. If a pharmaceutical company is trying to make a medicine, then they may be trying to discover certain medicines along the way. And for example, we got a grant for a medicine last year, and that medicine had activity against influenza and HIV. And so that's where we directed our care. Now, we knew at the time, this was a year ago, that it has activity against coronavirus, but it seemed very irrelevant. So we didn't apply for that. But since then, obviously, it's very relevant. And so now this drug that was already made is now in preclinical studies, which are essentially is research in animals. So vaccines for COVID-19 don't have that luxury because it's a novel virus. And so you have to start from scratch. So pharmaceutical companies are making vaccines. And so the research has already started in this early part of the process. Then what happens when a pharmaceutical company has a vaccine that they're making and they've tested it in preclinical studies and mice and ferrets and whatever, then they'll apply to the FDA for that, and it'll go through human trials, which is where you get the phase 1, phase 2, and phase 3. So after that, you'll have the process go through the phase 3. It'll go through another review phase with the FDA, and if it's favorable, then the FDA will approve it. So what I want you to notice is on the left side of this screen is how many studies and drugs and vaccines basically that there are. And as each process goes along, it gets narrowed down one after the other until there's not many left, basically. So here is what a pharmaceutical company has to include in their investigational new drug application before their first human patient. And that is information about the vaccine, how it is manufactured, the quality control test for the release, the vaccine safety, the data on animal testing, and the ability of the vaccine to elicit an immune response. This is something called immunogenicity, and basically says how well it works. Does it prevent the infection or does it not? And finally, the proposed clinical protocol for human studies, which includes all of the information like inclusion criteria, exclusion criteria, what blood tests are we going to get up front, and how often are people going to be monitored, what is the dose, how many doses, that kind of very detailed and specific information in the protocol, usually very long documents. So phase 1 studies is a safety study, and that's why I've highlighted it as such. The main reason is to find out if the vaccine is safe in humans. Maybe it was fine in ferrets and mice, but we need to test it in humans. The people that volunteer for this study have to be healthy, and they're usually young. The duration is short, and maybe as short as just 1 week. It's a very short study in just a few people, and the emphasis is on safety, not the immunogenicity, which means it's not testing to see if it works, it's just testing to see if people can tolerate it. Now, it answers a couple questions. What are the most common and serious adverse events? Another question it aims to answer is, how is the vaccine metabolized and excreted? Does it go through the kidneys? Does it go through the liver? Does the liver process it fine, but then the enzymes go up? Or does the kidney process it fine, but then you have some renal dysfunction? So those are the types of answers they can get from a phase 1 study. Now, as you can imagine, and I think I know what happened, was after they came out with these names, phase 1, phase 2, phase 3, there was a drug or a vaccine, and perhaps the pharmaceutical company was a little timid toward giving it to 90 people. They said, why don't we do a smaller study first before we do the phase 1 study? And so that's probably where phase 0 studies came, phase 0 studies came in. So this is an exploratory study, and you might just give it in a dozen people before you give it to the 90 people in phase 1. So it's usually a small dose. So it's performed strictly for toleration, not for actual therapy. And if those dozen people do fine, then they'll expand it and give it to phase 1, where they might give it to 90 people. So then if you go through the phase 0 and the phase 1, then you can go to phase 2. And this is more to see how the vaccine works. It's an immunogenicity study. It's medium size. We've gone from a few dozen to a few hundred. Volunteers are not necessarily healthy. They may have, for example, if it's a drug and it's a blood pressure drug, these people are going to have high blood pressure to see if it comes down. So monitoring for adverse events may be over months or even years. They can monitor these people. The emphasis is on immunogenicity while still evaluating safety. Some of the questions it aims to answer is, does a certain drug work in people with a certain condition? For example, that blood pressure issue, does it actually lower their blood pressure? For a vaccine, does it work in people who are at risk for COVID-19? Do they not get COVID-19? So then you have a phase 3 study. And a phase 3 study is a large study, and that is what we're hoping to get here at U of L, several thousand patients. So one pharmaceutical company will have a multitude of sites, 20, 50, 70, maybe 100 sites around the world where they are administering the vaccine or placebo to see if it works. The size allows for more thorough investigation of safety and the immunogenicity. You have different populations. Volunteers are diverse in their health, in their risks, in their race, in their sex, etc. There's different doses given if you're talking about a medicine or even a vaccine. And the monitoring may be for years to determine long-term side effects. The emphasis is on statistical power. So the more people in the study, the easier it is to show that a difference is statistically significant, that it is real. So let me give you an example about vaccine efficacy. So if a vaccine is 100% effective, that means everybody that gets it and who is exposed to influenza, for example, does not come down with the infection. So influenza vaccines, unfortunately, are about 40 to 60% efficacious. And if you remember, about 4 or 5 years ago, there was one where they did not guess the influenza strain so well, and it protected about 25%. So the FDA has declared that COVID-19 vaccine, if they're going to approve it, needs to be 50% effective. Okay? So in Kentucky, we have approximately a 6% positivity rate, which means out of 100 people who are tested, 6 are positive right now. Now, if you look at those 6 people, if we had a vaccine and those 6 people got it and it was 50% effective, then half of them actually would not have come down with COVID-19, even though they got exposed. And obviously, if it was 100% effective, then nobody would have gotten the infection. So what is the only way to test the effectiveness of a vaccine? And that is to randomize the vaccine. We'll randomize between what choices? And we'll say randomize between vaccine or placebo. Because if you say that a vaccine is better, then you have to say that it's better than an alternative. And the alternative right now is nothing, because that's all we have. So when you have phase 3 studies for COVID-19 vaccines, and as well as on the other study, you're going to have some people who are given the vaccine and some people who are given placebo, and then the statistics will be on comparing those two groups and hopefully seeing that the COVID-19 vaccine group did not get infection as much as the placebo. So how do you eliminate bias? And that is with double blinding. We don't know, as researchers, who gets the vaccine and who doesn't. We may be tempted to say, well, she's pretty sick, I want her to be sure she gets the vaccine, or she has this risk factor. And you, as the volunteer, may be tempted to say, well, now I know that I got the vaccine, so when I feel this headache, it's definitely due to the vaccine, when actually it's not at all. So both those things are prevented by it being double-blinded. So what we have are randomized, double-blinded, placebo-controlled vaccine studies, and that's what we hope to have here. So now here we are in the midst of our vaccine trial and phase 3, and what these arrows represent is anywhere that the FDA can ask for certain information. So they can ask for additional information, they can ask for an additional study, maybe a side study to be done, and they can also just halt the study. Now, some of these can be scheduled. For example, there's one early in phase 3 on my example, because that's a very common time to have the FDA say, after 20 or 50 patients, we're gonna pause your study. You're not allowed to enroll for about 3 days while we review the preliminary data, and if everything looks good, then you can continue. And we had a study recently where we had that scheduled in, and the preliminary data looked good enough that they said, while we're reviewing the information, you can enroll. And so after the 3 days, we're either going to say continue or stop. And that's what they'll have the prerogative to do here. And they have that prerogative anytime they want. They can just visit and say, we need this information, and until we get it, the study is paused. And then if they come in and they don't find it favorable, they can just stop it, which has happened. And sometimes they stop it because the drug or the vaccine is working so well, they want the placebo arm to go ahead and get it. So vaccine labeling is also a part of the IND application. So proper labeling is required so that the pharmacist knows how to store the drug, prepare it, and dispense it. And I'm not talking about the label on the bottle, but this is a label, it's a whole manuscript that comes with the drug. It also allows and tells the healthcare worker giving the vaccine how to administer it, when to give it, who it's intended for, and such. So then, once you have all this information, it can be submitted to the FDA, and it's submitted as a new drug application. So when they take it, they don't just approve it or decline it, then they move it on to a third-party review. So there's a committee called Vaccines and Related Biological Products Advisory Committee, and it has a mnemonic that's too long to repeat. Anyway, it's a committee of non-FDA experts. So they move it over to these people who are scientists, physicians, biostatisticians, and a consumer representative. a consumer representative is a volunteer who is actually getting the vaccine. So this group is made up of people who have the highest interest of all the volunteers who are getting the vaccine, because at least one of them is actually getting it. So this third party then provides information back to the FDA, and they provide information about the safety and the efficacy, the two main things that have to that are most relevant. You want to know that it's safe, and you want to know that it works, because you don't want a vaccine that's just safe that doesn't do anything, and you don't want a vaccine that does something but isn't safe. You want it to be both ways. So now we have a vaccine that's moved, and the FDA can take that information and hopefully approve it. And you see, by the time it gets to this point, there's not very many vaccines left. So after that, we move on to a phase 4. So right now, as of the other day, these little jars represent the number of vaccines. And you see the preclinical studies, there's the most. And then there's phase 1, there's a few there. Phase 2 has about a dozen, and right now there's 7 active phase 3 trials of COVID-19 vaccines, and then approved. This is for the United States, not Russia. You have none. So that's why there's so much interest in getting one in the United States. Now, these colors also represent something. You see that there is kind of a turquoise color that says virus. Those are 3 types of phase 3 vaccines that are attenuated or weakened virus. There's 2 that are purple, and those represent virus vectored vaccines. That's the kind, if you remember Dr. Ramirez's talk from last week when he talked about DNA or RNA of the coronavirus getting into our cells so it can reproduce the spike proteins or some other protein, so that once we make it, we can expose it to our antigen presenting cells, which can take it to our T helper cells. And that information can then be moved onto our immune system so that B cells can make antibodies, and that NK cells make killer cells that would kill any cell that's infected with coronavirus. So in this aspect, for example, adenovirus is the means by which they put the DNA or RNA in the adenovirus, put it in the person, and it gets into the human cell. And the last type is the rose-colored, and that says nucleic acid. Those are 2 new types of methods for vaccines that Dr. Ramirez went over last week, but it's the same concept of getting DNA or RNA into the cell. In one example, there's actually an electric impulse that moves it into the cell, and in another one where there's a micelle or a fat cell. And so it's kind of like the Trojan horse, where the DNA or RNA is inside that, and it allows it to get inside the cell so that we can generate just the protein parts, not the bad part of the coronavirus, and beef up our immune system so that if we ever get truly exposed, we have seen it before and we can mount a response. So I talked about the FDA continuing to monitor as the studies go on. They monitor vaccines and the manufacturing process to ensure continued safety. They can also do facility inspections wherever the vaccine is made, and they can request the manufacturer's own test for potency, safety, and purity of each vaccine lot. So pharmaceutical companies do their own studies because they want to have the best vaccine, because they're competing with other pharmaceutical companies. So in doing so, they generate their own data, and the FDA can request that data. Well, the other thing they can request is just samples of vaccine lots for themselves and do more research themselves. The FDA can see if the data they're getting matches what they've generated themselves. So a phase 4 study is done after a vaccine is on the market. And the studies involve thousands of the general population. And because it's so many, more people mean more adverse events may be revealed. There is something called a vaccine adverse event reporting system. And so your physician, if you ever have a side effect to a vaccine or a drug, it reports to this so that there's a giant database. And the duration of phase 4 studies may last for years because it's not holding anything up. It's already on the market. So now I want to talk a moment about Operation Warp Speed. The previous vaccine record, well, you know, every year they come up with an influenza vaccine. So that's generated in just under a year. But probably the record is in 2009 when we had H1N1, and that was in April, and they had a vaccine by September. So if you're talking about a new bacteria or virus that they've had to start from scratch, then mumps gets the award at 4 years from 1963, and then it was released in 1967. I think what's going to take the longest record is HIV, which is 36 years and counting as of right now. So the U.S. government is also operating and funding $9.5 billion to the vaccine industries to generate a COVID-19 vaccine. And vaccines do not usually get this kind of funding, and never before, actually. And so what the U.S. government has done is taken on the liability of making a vaccine that may not work. So with that liability on the U.S. government instead of the pharmaceutical industry, they're able to garner a lot of different resources and move forward much quickly than they ever have before. So there's also new methods that do not rely on growing whole viruses in chicken eggs like we did with old influenza vaccines. That takes a while, and we just skip that part. Now we have newer methods of driving the DNA or RNA into cells faster. And another method that's rather slower is generating viral proteins for vaccines. And then for this COVID-19, there's simply going to be less time in between study phases, whereas previously they may have waited between phase 1 and 2 and then 2 and 3, and there's nothing been so pressing as COVID-19. So between all those things that allow time to be saved, they've titled it Operation Warp Speed. So at this time, we'd like to answer your questions, and it looks like you're already starting to use the chat box. So just keep doing that, and we'll try to come up with answers between the two of us. Thank you for your time.
Very good. Thank you, Forrest. Well, I have a couple of questions for you, but I noticed that there's a question, if we know what type of vaccine we may have the possibility to study here in Louisville. And in reality, Forrest mentioned that these are usually combinations of the government working with some companies, and we were asked, since we are still in negotiation, we were asked to do not mention anything regarding this possible collaboration. Then we are, as part of our signing all this preliminary work, has been the idea that we're not supposed to discuss whatever we sign. Then I'm sorry that now this may change next week. We hope that next week we may, but at this moment we cannot discuss. This is why these presentations are for all the vaccines, all the phases. Of course, no question that there is one of these 7 vaccines that is in phase 3, but because it's, phase 3 is what we are going to be doing here. Now, can
I just say a comment
yes,
on that?
Yes.
If we revealed at this moment what the mechanism of the vaccine is, then there's so few that have gotten this far that it would be easy to identify which one it is. And then another way of Operation Warp Speed to speed things up would be to skip phase 3, which is what Russia did. They went from phase 2 to approve. And obviously we're not going to do that either. So hopefully we'll have more information soon.
Yes. Then there are a couple of questions, even with some names of the vaccines that are in the, then again, we are not going to be able to answer this one. But there is one question with the known mutations, but the virus is mutated. Do you think that the vaccine will need different vaccines? What do you think? What will be the comments of the mutations and vaccines?
Oh, I see. So MMR is an example of a vaccine where you get it, and then maybe you have a booster when you're about 18 and you're good for life. Whereas influenza is one that changes, and then you have to have one seasonally. So you're asking which one this is, and because it's novel, we don't know. I can only hope that it's one that we don't need every year, but maybe we'll be getting our flu shots and coronavirus vaccines every year. Who knows?
Yeah, then there's another comment that if the mutation will make the virus similar to the common cold. It's interesting. And I understand the question. We know the coronavirus now, the COVID-19. The virus is mutating, and the virus can mutate into a more virulent virus, or the virus can mutate into a less virulent virus. Then if the virus mutates into a more virulent virus, so the virus keeps changing, the vaccine, we may need, as Forrest mentioned, one vaccine every year. On the other hand, if the virus mutate to become less and less virulent, we may end up with this virus that causes like a common cold. These are all things that are in the literature, and we don't know what is going to happen. The problem for the vaccine is the virus mutates a lot, as Forrest mentioned, as the influenza virus mutate, then we may need to keep getting a new vaccine every year. Then every year when we go to the doctor, you're going to get the flu vaccine and the COVID-19 vaccine. This is a possibility. We don't know. There's a question, when the participants will know if they get the vaccine or placebo? You want to answer this one or?
Okay, it's gonna vary with the vaccine, but we don't know.
Yeah.
And the checks that the FDA does, that is even perhaps before it's finished, may change things altogether. But and we talked last week, maybe you weren't on the call, that if they find it to be effective and the study has finished, then at some point, you're right, they're going to reveal that so that the people who are in the placebo arm can go and get a vaccine.
Yes, because yes, then as Forrest mentioned, the study can be stopped when the FDA review the data, because let's say that it's already 500 individuals, and because the vaccine may not work, and then stop earlier, or may be stopped because the vaccine works much better than expected, and then you don't need so many patients and may stop earlier. But traditionally, usually when the study finished, they said the FDA now want a vaccine that at least is 50% effective. Then you have so many patients, you reach the 50% effectiveness, the vaccine is going to get approved, and this is the moment that everybody knows who got the vaccine, who got placebo, and everybody's invited to get the vaccine if you were in the placebo arm. There were a couple of questions regarding follow-up, and still, all the protocols of the vaccine have the same, more or less the same follow-up. You are going to be obtaining blood because, as Forrest mentioned, it's going to be important to see the immune response or the immunity that you develop, to see if the antibody response protect. But at the end of the day, the most important answer is not going to be so much how much antibodies or what level of antibodies. The answer on these phase 3s, do you get COVID or not? Mm-hmm. Does the vaccine protect against COVID or not?
And also, you may be asking about the follow-up, perhaps blood tests or nasopharyngeal swabs or that kind of thing. And so the protocol will outline that on this week, like week 4 or 8 or 12 or whatever, it'll be very specific. So when we have that information, certainly it'll be provided up front before you even sign a consent, so that you know how often people will be testing and that kind of thing. I think somebody asked where in Louisville will it be administered, and
I may say that we have to. All these things are we are still evaluating, because this will be just discussing specifically the study itself. And at this moment, we don't want to discuss the study itself. We want to discuss general concept of vaccines. This vaccine, like any other vaccine, will have to be refrigerated. It's going to be in a special place, and then usually you're going to have one site. But this is going to be, if we get the protocol moving forward, everybody's going to be known. And then there was a question on how do I know if I'm going to be selected for the vaccine. Again, based on the protocol, the protocols, as Forrest mentioned, is going to have inclusion criteria, exclusion criteria, and then based on the members, healthcare workers that are already in our register, we are going to let you know, okay, based on the criteria for the protocol, this is the group, this is going to be the first group, second group, third group. But I would like to discuss this more in detail if we have been, if we move forward with the study. It's an interesting question. If I get the immunization and then I get sick, will be positive if you are tested. Will COVID-19 positive or false positive due to immunization? We discussed that at this moment, antibody testing is not, we don't do antibody testing to see you have COVID or not. We do the nasopharyngeal swab, PCR, saliva PCR. It's usually through detection of the virus. Then if you have a vaccine, of course, the PCR is going to be negative. And now, for any vaccine, if someone decided to check your antibody levels, we hope that if you receive the vaccine, your antibody levels are going to be positive. Then this is going to be part of the discussion. Anyone that gets signed for a vaccine, then you know that you get antibodies, your antibody levels are going to be positive. As Forrest mentioned, we've been working a lot in an HIV vaccine, and this has been an issue. You are in an HIV vaccine because they give you, because you develop antibodies, and then your HIV test is going to be positive. There's always the consideration that your serology is going to be, we hope that it's going to be positive once you get the vaccine.
Right. Somebody asked, when will they be notified to be in the study? And, you know, we hope within a week, but we just don't know, and we don't have control of that. So maybe a few weeks. Somebody else asked, where on the body do you get the vaccine? And if it's not in the deltoid, then it would surprise me, but we'll have to wait to see the protocol, and the protocols have many of those details in it. So as soon as we have that, we'll certainly be able to answer those type of questions more specifically. And then somebody asked about the specific route. And again, if it's a shot in the deltoid, then I guess that's what they're talking about. There have been influenza live virus, vaccines, but none of them that have moved this as far. I showed those 7 that are in phase 3 right now are that type. And then somebody asked if the COVID test will affect the study or the results. So if you get tested for COVID-19, so that's just a PCR checking to see if it's there, and it's just going to say what status you are. So that's not going to affect the study or results, and probably it's going to be part of a test somewhere along the way while we're in the study, wouldn't you think?
Yes.
Getting antibodies in a nasopharyngeal swab. Somebody else asked if you have to be isolated at any point in the study, and I don't see any reason why anybody would have to be isolated, just like other vaccines where you generate antibodies, you don't have to be isolated. And since this is not a live virus, there shouldn't be any kind of virus even attenuated or weakened that you're shedding. So that wouldn't be a reason to isolate you either.
Yeah, it's very important. None of the 7 vaccines that are in phase 3 are a live attenuated vaccine. These are all essentially, there is no there's zero risk of getting COVID through the vaccine because there's no live attenuated vaccine. Again, there is a lot of questions, where the study is going to be done, what laboratories are going to be performed, compensation. As a matter of fact, we don't, and again, we are working with one company that we are close. We are in conversation with other companies. Then our goal will be if we can bring several trials to Louisville, and then it's going to be probably different protocols. But this is why I say at this moment, these are details that until we sign and start enrolling, and these all are details that are coming in the informed consent. Now, the informed consent has to be very detailed regarding these points.
Somebody was asking if this another phase would move this out into the community, and certainly that would be ultimately the goal of what this would be. And certainly, even if it would be approved early, then it would just be generated and made available to the public. Remember that this is open to healthcare workers in Kentuckiana. So although it's not open to students at schools, if there's a student nurse, then by all means, that could be somebody that's finding out more information and getting this because it's a healthcare worker.
Essentially, as Forrest mentioned, at this moment, in the general, probably the number in Kentucky is 6% of the number of positive individuals in our community. But we know that the number for healthcare workers that the CDC have discussed in the United States and some of the numbers is close to 12%. Then we as healthcare workers, we are in contact with patients. We have doubled. Some people say that our risk for being in contact with patients triple compared with the general population. Then this is where we are focused because I've been receiving a couple of also emails because we're focused on healthcare workers that are going to be in contact with patients. Not only because the risk double or triple, it's also, as Forrest mentioned, because we need to prove that the vaccines work, and if you have the higher risk individuals, in this case healthcare workers, then you are going to be able to prove the statistical significant difference between the two groups in a shorter period of time. Then if you go to a general population, the vaccine, you may need 1 year to find the difference. You go to healthcare workers, you may need only 6 months to find the difference. Then we just happen to be that for this type of, for COVID-19, if we are seeing patients, we just happen to be the one at a higher risk. And we're not saying treating patients, just being in contact with patients.
Somebody asked about reconstitution of the vaccine, and that'll depend on which vaccine. But again, I'll reiterate that, we actually hope to be the site for not just a vaccine, but maybe 2, 3, or even 4. So the other variable here is how many each one will want. We don't know if they're going to want 400 volunteers or 2,000. And if there's more than one, are we going to have 6,000 slots? So we hope to offer a spot for everybody. But if we don't have that, we will be notifying by lottery, and we'll certainly notify you one way or the other, especially when the quote is full. But the more we get, the more people we can vaccinate.
There's the idea of other groups, for instance, members of the community working in public schools, and these apply to essentially all the vaccine trials that are at this moment, as Forrest mentioned, in the phase 3. Everybody is looking for members of the community that are considered are high risk. And at this moment, it's clear that they are the healthcare workers because we are in contact with patients, and then members of the community that are regularly in contact with other individuals of the community. Then there's no question that teachers will be a risk if the schools are open. Not then if we are going to be in contact with kids, yes, because they are members of the community, or with adolescents. Then we have, but then we know that the bus drivers, for instance, are considered a risk. Anyone that is in contact with the communities is a risk. The higher risk is usually in contact with patients, but now we are learning that probably the difficulty in containing this pandemic in part is due to the 20-30% of the individuals with COVID-19 that are asymptomatic and they are able to transmit the disease. Then we know that as high as 1 in 3 persons with COVID-19 may have 2-3 weeks that they have no fever, no sore throat, they are completely asymptomatic, they have the virus in the oropharynx and they transmit the disease. And this is why we think that happened, that even though we are doing, we try to control, this has been so difficult to control. In other pandemics, with other infections, usually you are highly infectious when you have disease. In the case of influenza, we can transmit influenza 1 day before we develop fever, and then once, meanwhile, you have fever. Then it's easy to say, okay, this person with fever may be able to transmit the disease. With COVID-19, with all the large number of asymptomatic individuals, we may have a person in the community or a person in the hospital that we're not aware, and the person is admitted to the hospital without fever, without any problem, is admitted to the hospital because of uncontrolled diabetes, and without knowing he's carrying the virus for the next 7 days. And this is the risk, even though we try to protect ourselves, but there's always a risk of the asymptomatic person.
Very good. Somebody asked about adverse reactions in phase 3. Have they been mild? And right now they have not published preliminary data, but at least in phase 1 and 2, and phase 2 is the larger of those two studies, a vaccine wouldn't move from phase 1 to phase 2 unless it didn't have many adverse reactions. And it also, again, from phase 2 to phase 3. So they don't even get that far if they've got significant adverse reactions. With the larger size that a phase 3 study is, there may be some that turn up. And then, of course, phase 4, after it's released, when there's thousands upon thousands of people enrolled and there's the vaccine registry, then sometimes we find out about adverse reactions. So maybe you've been familiar when there's a drug that's been put on the market and then it gets pulled because once there was enough patients who got exposed and they realized there's been a problem with it. So as far as we know, we don't have a vaccine that's active right now that has a lot of adverse reactions just because it's made it that far. And that the FDA would not approve that type of thing. So the safety and efficacy is what they're looking at.
And just again, yes, just to clarify that phase 3, they just started all this month, or they're going to start next month. Then when someone asks, have we find any adverse events in phase 3? Well, we still don't have phase 3 studies. They are all starting really globally. They are all starting at this moment. But we know that based on the components of the vaccine, at least it is going to be unlikely that we're going to see severe adverse reactions because, for instance, for the, as Forrest mentioned, when you get a virus that you put the genetic material and inject the virus, this virus and adenovirus, and there are other studies done with this, and then you inject the nucleic acid, then people don't expect that these small pieces of genetic material of the virus is going to generate, too much of an adverse reaction. But it's something new, we don't know. But when we give an antibiotic that's going to act in a bacteria, and when you give a drug, you can see that that we may have problems with the kidney, the liver. Nobody expect any serious adverse reaction with this vaccine. The challenge is going to be the vaccine develop antibodies, and for how long you're going to have the antibodies.
Right.
There's a question if all these have been recorded. And yes, we recorded last Thursday. We are recording this Thursday discussion, and probably we're going to resend an email to everybody regarding where this information is going to be located. I have to tell you that we would like to communicate with all the members that register in this for interest more back and forth, but every form of communication that we have with any one of you has to go through the IRB or the university to be sure that even though you are not participating in any research, this is just a register to look at your interests, any form of communication first needs to be approved by the IRB to be sure that we don't misinform anyone with our communications. But definitely in the next one, we are going to give you the information where all these presentations are available.
It looks like Dr. Carrico posted in the chat box the links, but we'll still email it out. I think that's important for everybody to see.
Yeah, it's already there then. Yeah, because these are open discussion, then it's there for people, and I understand that CERID Louisville.org, Louisville COVID-19 vaccine study. Okay, very good. Good. And then at this point, if you have to guess how many months do you think to have a vaccine in the market, or how long? Because we started at phase 3, you say that then we need to analyze, then it has to go to the FDA, FDA has to approve. Since this is all supposed to be at the light speed.
Yeah.
Then
how long do you think? I think it'll take?
How long? Because one thing is for sure, this vaccine, regardless of what happened, Forrest and myself, we cannot take the vaccine because we are investigators. One of the exclusion criteria of every research that investigator cannot participate. Then we have to wait until the vaccine get approved. Then Forrest, when do you think that we can get the vaccine? When the vaccine is going to be FDA approved?
Well, I think that the FDA works at the same speed as the IRB. And if I were to grade our IRB for this year, 2020, I would give them an A+, because everything that we've put through a lot of studies. They have responded, they have worked outside of their normal hours. And it seems to me that the FDA right now with the COVID-19 vaccine has that same mindset where it's a priority. And so they're going to work at a speed that we've never appreciated before and we're not used to. So I would like to think, I'll just go out on a limb here because I'm getting used to guessing and being wrong. I'll just say February 14th.
Okay.
The day of love, we'll have our vaccine. So now it's recorded, right?
Yes, recorded.
Okay.
Then we're not going to be able to be part of this study or any other study that we're going to be investigators, but according with Forrest, I just need to survive until February 14th, and then I'm going to be safe.
Right.
No, but I agree. The idea was in an advisory board for an antibiotic that was submitted to to the FDA. And the challenge is that now everybody in the FDA is working on COVID. Then when other things are going to be approved, we don't know. But yes, and everybody is collaborating. This tremendous amount of collaboration. Do we have? Okay, we have another. Yes, it's excellent news for the IRB. Yes, as a matter of fact, to be even more detailed, in February, I think it was in February when we knew that by the end of February we knew that this was coming, and then the question was that then we have in March the idea that everybody's supposed to go home from the university and everybody's supposed to work from home. I think we went with Ruth Carrico to the IRB and discussed that we're applying all our initial protocols. And one of our protocols was exactly to study healthcare workers. These have been always our primary interest because we know that we are part of the solution, but also part of the problem here because we were coming with COVID-19. And from the very beginning, the IRB mentioned that they will work regular hours or extra hours, and they will always look at our proposals for COVID-19 in a very special way. Then everybody, I think, understood from every aspect that this is a pandemic and we need to do a business not as usual. Then we are very happy. We are also, for instance, at this moment in this register, I don't know if any one of our group have the number of healthcare workers that have been in our register, but when we look at this, we started with this one Friday, but Monday there were 100 people registered. Then next week we were 2,000, and the number of healthcare workers interested in helping in developing, in moving forward this vaccine keep expanding. But at the same time, these numbers close to 3,000 now, and I don't know what is the number today. But this number has been also very helpful for us to discuss with companies to say that in the Kentuckiana region, we already have 3,000 healthcare workers that are interested to participate. Then this is helping us also to when we discuss with other companies, because again, there is always a competition because everybody wants to have the vaccine, and the competition is what is your track record, how many healthcare workers are interested, and also the companies are looking at what happened, what are the hot spots for the disease at this moment. Then they say that they think, well, we would like to start vaccinated the third week of September. They're going to be looking in all these different sites that they have all over the world. Okay, what happened in September, October, November, to see that, as we discussed, what are going to be the areas where people are going to be at high risk, because these are the areas that they want to activate and start doing the vaccine trial as soon as possible. Then there are a lot of factors here that are outside of our control, but we'll let you know as soon as possible. We are activated to start enrolling, and of course the people that are registered are going to be the first one to know, because this is why we have this communication with you. We have your email to be sure that as soon as we know, next day you are going to know and you get the information. There's a question if we will know the result of our antibody lab work. Yes, in reality, we are not going to, nobody is going to know the result of the antibodies lab work, because this will be, as Forrest mentioned, it's very important that the study is randomized. You don't know if you get the vaccine or you get the placebo, but it's also very important that the study is blinded, meaning that I don't know when I talk to any one of you, if you got the vaccine or placebo, and you don't know if you got the vaccine or placebo. Because another thing that we partially discussed last time at this part of this, any vaccine study, is that vaccines, there are two ways that we think that the vaccines are going to work. If you get high level of antibodies, you may be completely protected. Then you don't get COVID. But some of us may develop certain level of antibodies and may not be completely protected, but may give you get COVID, you're going to get a mild COVID. Then you may get COVID and you're going to stay home, but you don't need to be hospitalized. Or you get COVID and you don't get pneumonia. Then even if you have the vaccine, you may still get COVID, but a mild form of COVID. If you get the vaccine, we know that at 30 days, based on the animal studies and phase 1, phase 2 studies, we know that at 30 days, almost 100% of the individuals in phase 1, phase 2 that got the vaccine are going to have antibodies. Then if you draw blood and in 30 days, you know that you have antibodies, then you know that you got the vaccine. And if I know that you have antibodies, then I know that you got the vaccine, then the blinded is no longer there. Then the idea is that to be sure that everybody is blinded, you're not going to get the results of the laboratory test until the study is finalized.
So if you made antibodies, that would kind of give it away that you probably got the vaccine.
Yes. If multiple vaccines are tested, then the placebo group will be valid for multiple vaccines. This is an important consideration. Yes. And really, the protocols, as we mentioned, as far as already alluded, you may have one group that is the placebo. This may be one group. But then, as far as mentioned, you may have receiving the vaccine, you may have two different groups. One dose of the vaccine and the other a higher dose. Or one group will get one dose of the vaccine and the other group will get two doses of the vaccine. Then it's not going to be just vaccine placebo. Most likely protocols in phase 3 are going to be learning other things just besides the vaccine. You start first placebo and vaccine in healthy individuals, and then you are going to go probably to individuals that are going to be more than 60, and then you want to see how much of the vaccine, for instance, happening with the flu vaccine. You now have the high dose flu that is indicated probably for the elderly, because we are more difficult to develop antibodies. Then you get a higher dose of the flu vaccine. I'm sure the same is going to happen with the COVID. We're going to have different doses trying to optimize the result. Then it's going to be different placebo for healthy individuals, placebo for elderly, placebos with individuals with comorbidities, placebos with individuals that are immunocompromised, because this vaccine, the time will arrive that we're going to give the vaccine in patients that are taking biologic, patients with cancer. Then it's going to be a lot of placebo groups that, yes, once you have a placebo, then multiple vaccines can use the same placebo. I agree.
And the arms may not be a 1-to-1 ratio either. It may be two people get a vaccine and there's one in the placebo. We'll just have to see the study. And then somebody asked if, let's see.
If someone has already
can you posted, get it again?
Yes, if someone has already posted, you feel that you can get it again. And
I know there's some articles about that, but I'm skeptical as an infection doctor.
Well, the answer to your question is we still don't know.
Right.
The answer to your question is that we know that there's a group of when you get the infection, we know that the level of antibodies that we develop after COVID-19, some patients with very mild COVID, they don't develop antibodies. Some patients with severe COVID, they develop high level of antibodies. But still, the question is, we don't know This is from the total antibodies, but then this is what is described as the neutralizing antibodies or the blocking antibodies. There is a subset of the antibodies that are more protected. And some people have a lot of neutralizing antibodies, others develop almost no neutralizing antibodies. Then the concept at this moment is that because you have, for instance, when you get the antibody with a drop from the drop of blood, this definitely tell you antibodies, yes, no. For these studies, you really need to figure out what type of antibodies. Are you having blocking antibodies? And as Forrest alluded also, it's not just the antibodies. We need to look at cells. We need to look at the B cells. You need to look at the T cells. This is why to get a real answer of your immune response, you need to have a blood test. And then if the protocol is going to accept individuals that when we draw blood, the antibodies are positive, and then you have, because you have the infection, let's say 2 months ago, this is going to be dependent of the protocol. Then, as Forrest mentioned, the protocol is going to have some exclusion criteria. We don't know if some protocols may say, we are going to draw blood. If you already have antibodies, then you are not going to be a candidate for the vaccine, or yes, you may be a candidate for the vaccine or to be part of the study. We don't know this answer. The answer to, if I get COVID, can I get COVID again? We still don't know the answer, but it seems to be that some patients with COVID have not developed antibodies, or in some patients, antibodies tend to decrease very rapidly. The concept is that at this moment, there is a possibility. At this moment, if you have COVID, nobody will say, okay, don't use protection anymore because you already have COVID. We will say to any healthcare worker, keep using full protection because we don't know, and you may still get COVID again at this moment. Then it's 5 o'clock. We thank you very much for all your comments, questions. And then this coming Thursday, we are going to be discussing safety. Then Ruth Carrico is going to review everything that we know on safety until Wednesday evening. And then Thursday, we discuss safety of the vaccines. Thank you for your attendance.
Yes, and thank you for your interest.
Thank you very much.
Bye-bye.